This video will walk through the steps required to upload personal data files for analysis in PMART. Currently, PMART supports the analysis of peptide, protein, and gene level data. Additional types of data will be supported in the near future. In this example, we will be uploading peptide level data that includes a mapping to both proteins and genes. More information on file format requirements, including example files, is available in writing here or as a video on our YouTube channel. To begin, we select Upload Personal Dataset Files. Now we're prompted to upload our data files and specify properties about our data. The first file to upload is the expression data file. This contains the numeric abundance data associated with each sample. Click on Choose to select a file. For a preview of your data, you can click on Data Preview. Now we're prompted to select the data type we've uploaded, in our case peptide. The missing value symbol. In our file, missing values are denoted by an NA. Since we've uploaded peptide data, we have the option to quantitate to either the protein or the gene level if we would like, and if we provide a mapping to those levels. If we do not have a mapping to those levels or do not wish to quantitate, we can select no quantitation. For our example, we'll select to quantitate to the protein level. Next, we must specify whether the data has already been normalized or not. Ours has not been normalized. Next, we must select the unique identifier for the peptides. The drop-down provides the column names for the data file that we have uploaded. In our case, all of the numeric values are the sample names, and our peptide identifier is located in the peptide column. Finally, we must select the scale of our data. We have uploaded raw data. It is not on the log scale, so we select raw. Because the data is not on the log scale already, we must choose how we'd like to transform it. We will select log 2. Next, we're prompted to upload the sample information file, which contains information about samples that might be used for the downstream analyses. First, we must choose the sample identifier, which is the column in our data that identifies the samples uniquely. The drop-down provides a list of all of the column names in the file we have uploaded. In our case, the sample identifier is called sample ID. Next, we must select the clinical features of interest. These are the columns in the sample information file that we would like to have the option of using in downstream statistical analyses. For this example, we will select initial pathologic diagnosis method and vital status. To select multiple clinical features of interest, hold down Shift and click on the features. Finally, we have the option of uploading a metadata information file. This is the file that contains the mapping from peptides to proteins and genes. For this file, we must select a mapping type. We'll choose protein as our mapping type 1, and an identifier that is associated with that mapping type. The drop-down gives the column names in the metadata information file, and we must select the column name that identifies the mapping type that we've selected. In this case, it is called protein. We also have the option of selecting a second mapping type. Since we chose earlier to quantitate to the protein level, the second mapping type is not required but will be carried through for downstream visualization. We will select gene for our second mapping type. Layer number 2 specifies the column name and the metadata information file associated with that second mapping type. In this case, it is gene. Now that we have uploaded our data files and provided the required information, we can click Analyze Data to proceed with PMART. On this page, we are prompted to select a main effect from the clinical variables of interest that we specified previously. The main effect will be used to make the statistical comparisons going forward. For our example, we will select Initial Pathologic Diagnosis Method. We now have the option of selecting a second main effect for our analysis. Since we had only selected 2 clinical features of interest on the previous page, and we've already chosen Initial Pathologic Diagnosis Method as our first main effect, we are left with the choice of vital status for a second main effect. However, for our example, we will proceed with the single main effect. The next step is to select the analysis modules. More information on this and navigating through PMART can be found in the PMART Cancer Introduction video on our YouTube channel.